All right. Hello and good morning or good evening to everyone. Before I start off with this presentation, though, I wanted to ask you all a quick question. How much do you really know about your eyes? Before you shrug it off, think about it. Our eyes take in light every second of the day, reflecting off trillions of particles around you. And our brain translates those trillions of waves into an image, lets you recognize each object in that image, and allows you to react to all of your surroundings. And this is all done within a millisecond. But pure light doesn't really carry any information. Pure light just moves around freely in an abstract way. How do our eyes combine those meaningless waves and weave them into an image of the world that we see, literally controlling and manipulating light molecules around us? Hopefully you keep this question in the back of your mind during this talk, and by the end, I hope to open your eyes to what science has revealed and also what it hasn't. For my project, I wrote a review paper on retinal ganglion cells, one of the fundamental neurons in the eye, and how their development affect our eyes, how our eyes develop, and how neural systems develop as well. And I wanted to share everything I learned while writing this paper with you. I'm going to begin with a basic introduction to the retina, then the processes conducted in the retina, and then move into retinal progenitor cells, retinal ganglion cells, and different neural systems. So to introduce you to the retina, the best way is to just imagine your eye working right now, watching this presentation. Light is currently reflecting off your screen and hitting the back of your eye, which is a light-sensitive film called the retina. The retina contains retinal ganglion cells, photoreceptor cells, amacrine, bipolar, glial, and horizontal cells. These cells are all part of the main retinal process called retinal phototransduction. This is the process of converting photons, or light particles, into images. Each one of these cells has a separate function in this process. Photoreceptors are the cells that absorb light first, then retinal ganglion cells relay the photoreceptor signals into the brain. Amacrine, bipolar, horizontal, and glial cells connect and regulate retinal ganglion cells in different ways, keeping the retina healthy and neural pathways working. Now, before I move into the details of how each cell works and how they develop, I want to leave you with this fascinating question about the process of retinal phototransduction. Light is constantly entering the eye from all directions, reflecting off hundreds of objects. But the light particles reflected from an object 100 feet away are entering your eye at the same time as light particles from objects 1 foot away. So how does the brain know to produce an image of something 100 feet away and something 1 foot away? Light doesn't carry information about how far it's traveled to get to your eye. So how is this possible? The answer is we don't actually have an answer right now. It's one of the most interesting and unique unanswered questions in all of neuroscience, and researchers work towards an explanation every day. All right, now you have a basic understanding of how the retina and retinal phototransduction works. I want to move into the structure and how the cells in the retina develop. And this all starts with retinal progenitor cells. RPCs for short, the parent cells of the retina. The process of RPC development can get complicated, so let's just take one RPC from the start to the end of its journey and follow it. This little RPC we're watching needs to do 3 main things in its life. First, be created in areas called niches. Second, get pushed to the center of the retina to make room for more cells like it. And third, differentiate into a specific type of cell. Now the first step, where RPCs originate. They develop in niches, which are basically just areas in the retina where stem cells can be created. The main niche in the retina is called the ciliary marginal zone. Cells in the ciliary marginal zone duplicate and push each other to the center of the retina. Okay, so now we followed the creation of this RPC in the ciliary marginal zone and how it's been pushed centrally. Once this happens, they differentiate into different cells like retinal ganglion cells, amacrine cells, etc. What controls this differentiation, and when do they choose to differentiate? Well, in the past, it was thought that the molecules inside and outside the cell are kind of preset to induce differentiation once the cell was in the right spot. But it was actually recently found that RPCs differentiate probabilistically. This means that there's a set probability for RPCs to differentiate into a certain type of cell. All right, so to recap, this little retinal progenitor cell we followed has been created, pushed to the center of the retina, and differentiated. This differentiation is controlled through probability mechanisms. Now, let's say it differentiated and is now a retinal ganglion cell. At first, it's a baby. When a retinal ganglion cell is first formed, it's not born fully developed and ready to serve its function. This little cell now has to go through an extensive development process to become a fully functioning retinal ganglion cell. We're going to look at three main parts of its development: the development of its dendrites, its axon, and its synapses. Okay, so now let's look at the development of each of these three parts. Dendrites actually begin as neurites, little baby branches that grow unchecked all over the place. In the diagram on the slide here, the dendrites are the upper branches that reach into different cells. Throughout development, these branches are remodeled and pruned away in a process called dendrite remodeling. Dendrite remodeling is a careful, specific process that's controlled and guided by a number of molecules and transcription factors in the cell. Axon development is a bit more complicated, as there are many parts of the axon. First, the axon initial segment, which is kind of like the top of the trunk. This is a relatively unknown region of the axon, but there have been multiple new findings on how this develops in recent years. The axon initial segment contains a specialized organelle called the cisternal organelle. Its function has been somewhat of a mystery until recently, when it was found to actually control the length of the axon initial segment based on the amount of light available when the organism first opens its eyes. When the organism first opens its eyes, if it's in a very dark environment, the axon initial segment will actually lengthen itself to try to propagate stronger signals because there's no stimulation from light. The myelin sheath is another part of the axon that develops similar to dendrites as it remodels. It lengthens and shortens throughout development. Now the final stage of retinal ganglion cell development, synapses. Synapse development is very simple. The cell needs to form ion receptors, a synapse membrane, and molecular systems to develop chemicals in between the synapses. The formation of all of these is controlled by transcription factors in the cell. Okay, so now our little cell has gone through development and has a functioning axon, dendrite system, and a variety of synapses. And it's a fully functional retinal ganglion cell now. It has to now interact with multiple different cells around it, creating a complex and vivid system. The interactions between RGCs and glial, amacrine, and bipolar cells are incredibly important in maintaining the health of RGCs and regulating RGC signals too. Glial cells mainly deal with regulating RGC life cycles, and the other cells deal with regulating RGC inputs and outputs. Now, finally, at the largest scale, I wanted to look at how RGCs as a whole affect neural systems in the brain, specifically memory, defense response, and circadian rhythm. A study by Mardersteck and colleagues in 2017 aimed to find every retinorecipient region of the mouse brain, or every region of the brain that RGCs project to. Mice brains and retinal systems are incredibly similar to humans. The study found around 60 regions of the brain that RGCs project to, including the thalamus, suprachiasmatic nucleus, and nucleus reunions. These are the three regions that are important to the three systems we're examining today. For defense response, thalamic circuits take direct input from RGCs. This was proven by a recent study where certain types of RGCs were made defective in mice through the knockout of a gene. A regular defense response in mice is flight or freeze, where the mouse quickly decides to either run or freeze in its spot. However, in mice with defective RGCs, when presented in front of a looming and dangerous stimulus, the fleeing response was almost completely removed, interestingly, and the mice would simply just flee. The fleeing response was almost completely removed, and the mice would simply just freeze, confirming the role of RGCs in defense response. Circadian rhythm, or your body clock as many call it, is also heavily dependent on vision. Seeing daylight or other stimuli heavily affects your body's sleep cycles and appetite. The suprachiasmatic nucleus, a part of the brain that communicates directly to sleep hormone glands, also takes direct input from RGCs. Now lastly, memory is a system where the role of RGCs is largely a mystery. This connection needs to be studied far more. However, the nucleus reunions, a section of the thalamus that deals with spatial awareness and spatial memory, does take input from certain types of RGCs, and stimulating these RGCs does result in an increase in spatial memory. Now, before I open the floor to questions, I wanted to leave you with this. Understanding the retina and its processes, the development of the different types of cells in it, and the interactions between those cells and neural systems is incredibly important due to the implications in medicine. Studying the retina in a detailed way can be used to treat neurodegenerative diseases in the retina, such as diabetic retinopathy and more. Okay. Before I open the floor to questions, I wanted to thank you all for listening to my talk and give a special thanks to my mentor, Mr. Trevor Doctor. Hopefully throughout my presentation, you got to answer that question I first asked you at the beginning, and you leave today with a better understanding of how the retina and your eyes work. Thank you.
Thanks. I think Alexis had a question.
Okay. Actually, the development of the axon is probably the least understood part of retinal ganglion cell development right now. The axon initial segment, its development was only found last year, and the role of the cisternal organelle. So there needs to be a lot more research done on that, because the mechanisms behind how the axon initial segment starts creating action potentials in the cell is not really understood right now.
We can wait a couple more minutes as well, or maybe up to a minute for any other questions to come through the chat. I also had a question in the meantime. You mentioned this great mystery about like depth perception or how far away something is. Yeah. Do you have any guesses or ideas, or does the field have any guesses, or is this does this mean we have guesses but we don't have any answers, or does this mean we just don't even know what a guess would be for how this works?
So when I was writing my research paper, I did spend like a few days, not a few days, but like one day looking at this, because it's just a completely nebulous question. Nobody knows the answer. And I basically just found a bunch of guesses online, but no real evidence to back anything up. But I don't think anyone really has an answer right now, because light doesn't carry information about how far it's traveled to get to your eye. So it doesn't really make sense, but nobody really has a good explanation.
Cool, thanks so much. I think Steven's going to come back in a minute and add the next person, because I still don't have presenter controls. Moderator controls. But thanks so much, Rohit. Okay. The next presenter will be Krrishika. I guess you can just get ready in the meantime until Steven's able to add you in.
So, hello everyone, my name is Krrishika Saxena, and I'm currently a sophomore from Arcadia High School, California. So I'm going to introduce myself so you guys know a little bit about me before I present my research. So, some things about me. I absolutely love the medical field, and I wanted to be a doctor ever since I was a kid. Usually kids are scared to go to the doctor, but I was the opposite. I loved the way how they worked, and it just made I wanted to do the same when I was older. So from the medical field, I would say neuroscience is a topic that really fascinates me and interests me, just how the brain affects our body cognitively and physically, and how everything's dependent on the brain just amazes me. And that's why I chose to research about Alzheimer's, which is a brain disease. And yeah, let's get started. So I researched understanding Alzheimer's risk factors associated with the prevalence of U.S. populations and women. So first I'll be talking about Alzheimer's, the risks and prevalence, the therapeutic treatments, and then we can have our Q&A session. So what exactly is Alzheimer's? So I think many of you might have heard the name before, but not know what exactly it is. So Alzheimer's is a type of dementia. It accounts for 60 to 80% of cases, so most of them. It's basically an irreversible progressive brain disorder that gradually damages memory and mental capability. So someone with Alzheimer's would have trouble driving, eating, just doing the most basic tasks. So it gets severe as it advances. So worldwide numbers. So looking at these numbers, it was an eye-opener to me. Millions of people throughout the world and in America suffer from this disease. And I was just amazed. I was like, there is no cure yet. And by 2060, these numbers will for sure double. So I wanted to do my part. I wanted to research and educate people about the risks and treatments so they know early on. And even if I can help someone decrease the chances of getting Alzheimer's, my purpose would be fulfilled. So Dr. Alois Alzheimer's was the first one to discover this disease, and since then, many researchers are here to identify the risks and causes that can aid in the development of this cure. So the two main causes of the disease are amyloid beta accumulation and neurofibrillary tangles. So amyloid beta accumulation is basically an amyloid beta protein that clumps or forms around the neurons, which is the brain cell, causing them to die due to immune response. And neurofibrillary tangles are tangles inside the neurons that interfere with cell-to-cell communication or machinery, and which kills the neurons at the end. So I researched lots of risks for this, but I categorized them into three categories. So genetic, behavioral, environmental, and I'll be talking about one risk per category more in depth. So before I get started, one thing I noticed when I was researching is that Alzheimer's affects a certain population or women more than other people, and I think this is for every single disease. Like at least I've noticed that there's some diseases affect more populations than others. For Alzheimer's, it was African Americans had the most highest prevalence, then Hispanics, then non-Hispanic whites, American Indian, Alaskan Natives, and then lastly Asian and Pacific Islanders. And for this is the most shocking fact to me, that women are disproportionately affected by Alzheimer's than men. Out of the 5 million people living with Alzheimer's in the US, 3.2 million are women. So when I was researching the risk kind of explained why these individuals are more prevalent than others. So let's get started. So the risk and prevalence. So the genetic risk that I researched was ApoE4. So ApoE is a gene, and gene makes protein, and this gene makes the apoprotein E that aids in the transportation of cholesterol. There are 3 variants of this gene. You might have heard the word variant from Delta variant or COVID variant, so I think we're all familiar with this term. So 3 types are ApoE2, ApoE3, and ApoE4. ApoE2 seems to be protective against Alzheimer's due to a lower binding affinity for the low-density lipoprotein, which means less amyloid beta plaques around the neurons. ApoE3 does not increase or decrease. It's the most common and normal type of the ApoE, and ApoE4 actually increases the chances of one getting Alzheimer's, as it has a great effect on AB clearance and form the plaques around the neurons. Here are all the functions of the ApoE and the 3 types in the protein. So prevalence and ethnicity. So African Americans have the highest prevalence of the ApoE4 allele in the United States. So this could explain why African Americans have the most cases of Alzheimer's. So knowing this, I could connect it back to that. And ApoE3 was common between all U.S. ethnicities. And then for Asian Americans have the fewest cases of Alzheimer's disease, like I mentioned earlier. And then this may be due to because ApoE2 levels were found to be higher in Asian countries such as China, the Middle East, and others. So here's a map of all the ApoE alleles and the pairs. So in women are more affected by ApoE4 than men, which is kind of weird because men and women can both get ApoE4. They have equal chances. However, women tend to have worse memory performance, greater brain atrophy, and lower brain metabolism from this. And the ApoE gender bias may be explained by hormones and tau protein. So the behavioral risk factor I researched was sleep. So sleep is necessary for several brain activities, including neurotransmission. As we know, our brain is still functioning when we're asleep. This can explain why we have dreams. So during the day, lots of chemical or toxic clump up in our brain, and amyloid beta is one of them that clumps up. And when we sleep, the spinal fluid washes all these chemical toxins away. So lack of sleep or insomnia can raise beta amyloid levels in the brain, increasing the risk of Alzheimer's disease. So the prevalence in ethnicity. So one of the primary causes of sleep is stress, and the stress hormone is known to be cortisol. So high cortisol levels in individuals have been associated with an increased risk for dementia and Alzheimer's diseases. And then I found out Hispanics and Black on average had the highest amount of stress compared to white and others. They also have more stress than whites due to average cortisol level, epinephrine, and norepinephrine levels. And you can see by the images here the categories of why they're more stressed and each level. So this can explain why Hispanics and Black on average have more cases of Alzheimer's. And then women's sleep quality is usually worse than men. Many research had proven this. And hormones influence women's sleep pattern. For example, women experience hot flashes during menopause and sleep apnea, which can disrupt their sleep and increase the chances of getting Alzheimer's. For environmental risk, I studied poor education. So we all know education is really important for the brain. This is why, because it promotes neuroplasticity. So what neuroplasticity is, it's stronger neuron connections in the brain. So when you learn something, the neurons form stronger connections, especially when we're young. So not studying or obtaining sufficient education can lead to Alzheimer's, because in Alzheimer's, the opposite happens. Neurons break connections and neurons die. So having lack of education can speed Alzheimer's rate. And for prevalence and ethnicities, when compared to whites, Black had a substantially greater prevalence of dementia due to lack of education. And the same was for Hispanics and African Americans for cognitive impairment, which is a common aspect for Alzheimer's disease. Prevalence in women. So women compared to men, as we know, in the past have had less education opportunities. In a study, the incident rate for Alzheimer's disease was 8% in men and 1.4% in women. So, 0.8% in men and 1.4% in women. So lower education may have a more detrimental influence on women compared to men. And here's a data that shows education levels throughout the past century. So therapeutic treatments. So though there is not a cure yet, there is still hope. These treatments are really still effective even if there isn't a cure, because by the time a person realizes they have Alzheimer's, the plaques and neuropil retangles have been accumulating for the past 30 years. So it might be a little too late when you find out you have Alzheimer's. So this is why it's important to know the risks early on and the treatments associated with it. So three categories of these treatments. So I included two.
Do you think you can wrap it up in the next
Yeah,
slide?
sorry, this is the last slide. Sorry. So the two treatments for genetic risk are gene editing by CRISPR and anti-ApoE for immunotherapy. Two treatments for lack of sleep are trazodone and bright light therapy, and two treatments for lack of education are cognitive stimulation therapy and cognitive rehabilitation. So though there's not a cure, there is still hope. Researching and learning about Alzheimer's was an eye-opener to me, and I wanted to make an impact and educate as many as I could, and this symposium is a great opportunity to do so. So I could only reach so many people, so be sure to educate the people around you about the risks and treatment. Together, we can put an end to Alzheimer's. So that's it for now. Special thanks to my Polygence mentor, Sabine. I could have not done it without her, so I can take any questions I have.
All righty. Thanks, Krrishika. I think I'm going to lead the Q&A while Jen is going to add Noora, who is the next presenter. Alexis has one question in the chat. She says, this was excellent information. What do you think is one of the most exciting areas for further research on Alzheimer's?
So, there's like the two main causes was the amyloid beta and neurofiber triangle. There's so many more causes to this disease. So I think every day we're just finding more information about this stuff. There are many more causes to these. For another gene that causes Alzheimer's is the PS1 and PS2. So I think that it's exciting to find new causes every single day, and I think in the end we'll find a cure for all of these causes. Thank you. Is there any more questions?
I think you're all set. Thanks so much, Krrishika. Great presentation.
Thank you.
All right. Let me share my screen.
And then same thing for you. I'm going to leave, and then I will come back into the room at 6 minutes as your 2-minute warning, and then I'll turn on my video when it's time to wrap up.
Okay, thank you.
All right, thanks so much. Good luck.
Thank you. Hi, my name is Noora Mathur, and I will be presenting on the connection between maternal exposure to environmental insults and the development of congenital heart defects. First, a little about me. I was born with a congenital heart defect called Epstein's anomaly. This is a defect in the tricuspid valve between the right atrium and ventricle. Presumably, my interest in cardiology stemmed from this. In all my years of living and studying my defect, I have always wondered what caused it, which prompted me to begin this project. I'm a senior at Wilcox High School in Santa Clara, and this is my research. Congenital heart defects are birth defects which affect heart structure and function. They are the most common birth defect, with 40,000 infants born with congenital heart defects each year, and 2 to 3 million people living with heart defects in the US. There are multiple complications associated with heart defects, such as irregular heart rhythm, heart infection, stroke, hypertension, and heart failure. Patients are also more susceptible to respiratory diseases, such as pneumonia and COVID-19. The causes are largely genetic, but about 10% of birth defects are thought to be caused by environmental risk factors, which is what I focused on. Maternity is a sensitive time period in which the environmental risk factors have a serious impact on the health of the infant. Every part of the infant forms at specific stages, so exposure to harmful substances can affect proper development. The heart forms early in pregnancy and continues developing throughout the pregnancy, meaning it has a greater chance of being affected by environmental insults. The environmental risk factors I explored throughout my research were tobacco smoke, air pollution, water contamination, and alcohol consumption. Tobacco smoke has the highest correlation to the development of heart defects. The greatest risk occurs during the periconceptional period, or 3 months before pregnancy through the first trimester. Secondhand smoke exposure, such as paternal smoking, is known to cause conotruncal cardiac defects, septal defects, and ventricular outflow tract obstructions. Conotruncal cardiac defects are outflow defects that disrupt proper blood flow. Septal defects are a category of heart defects that alter the walls or valves of the heart. Ventricular outflow tract obstructions occur when the vessels or valves taking blood away from the ventricles swell and prevent the flow of blood. Direct maternal smoking during the periconceptional period contributes to the development of atrioventricular septal defect, or AVSD. This is a defect in which the valves between the right and left sides of the heart have not been formed properly. Smoking is significantly linked to the development of this particular defect, as 26% of mothers of AVSD patients report smoking during pregnancy. Water contamination also has a high correlation to the development of congenital heart defects. The information is dated, but a 1973 study found that 1/3 of patients with heart defects had lived in an area of Tucson Valley that held water contaminated with trichloroethylene. This chemical contains chlorine, which is dangerous to humans. It increased the chance of developing heart defects by 17%, with a 58% chance of ventricular septal defect, or VSD. This is a defect in which there is a hole in the interventricular septum, or the wall between the ventricles. This allows oxygenated blood to mix with deoxygenated blood and cause hypoxia, which interferes with heart and brain function. Though trichloroethylene was used widely as an anesthetic and industrial solvent, it was later banned in the US due to its toxicity and potential carcinogenic impact. Air pollution also has a correlation to the development of heart defects. Maternal exposure to air pollution in the first trimester posed the greatest risk and increased the risk of developing congenital heart defects by 40 to 70%. It has also been determined that exposure to ozone in particular increases the chance of developing VSD and tetralogy of Fallot, or TOF. Tetralogy of Fallot is a more serious defect which involves 4 congenital heart defects working in tandem. The first is VSD, the ventricular defect described earlier. The second is pulmonary valve stenosis, in which the pulmonary valve does not open and close properly. This interferes with oxygen delivery because this valve takes the blood to the lungs to be oxygenated. The third defect is ventricular hypertrophy, which can causes swelling in the wall of the right ventricle. The final defect that contributes to TOF is an aortic valve defect. It manifests as swelling and prevents the flow of blood in the correct direction. Tetralogy of Fallot most often requires surgery, and it can result in delayed development. Alcohol consumption was the final environmental insult I explored, which has a small correlation to congenital heart defects. 1 in 9 pregnant women consume alcohol, and of that, 1/3 of them participate in binge drinking. Binge drinking is defined as consuming 4 or more drinks on one occasion. Research has indicated that there is a small increase in risk for the development of tetralogy of Fallot due to alcohol consumption during the periconceptional period, which is 3 months before pregnancy through the first trimester, as mentioned earlier. In conclusion, congenital heart defects are a serious birth defect that require more research into their causes, with 1% of newborns affected every year in the US. Complications are lifelong, and causes are not well understood. Air pollution is linked to VSD and TOF, tobacco smoke to conotruncal defects and AVSD, water contamination to VSD, and alcohol consumption to TOF. However, this information is limited and somewhat outdated. With newer technologies available, more research into their causes and potential solutions should be conducted. For example, NASA has developed a heart monitor that allows cardiologists to watch their patients and collect data that could be useful for future research. Apple's Research Kit is an open source framework that allows medical researchers to create diagnostic apps. One such app helps diagnose heart disease. With such technology available, cardiac research should be progressed. I hope to galvanize further discussion around this important issue. Thank you, and special thanks to my mentor, Brianna Banks.
Thanks, Noora. That was great.
Thank you.
We have a little bit of time for some questions in the chat. Alexa.
Yeah, sorry. Yeah, our CHD studies were pretty outdated because all the research that I found, many of these studies are fairly dated. I believe the most recent one I found was on tobacco smoke, tobacco smoke research was very recent, with around 2018 or 2019, but many of the other things I found were not recent at all. As I said, the water contamination was based on a 1973 study. The alcohol consumption was 1984, and then the next thing that I found about it was 2012, and there isn't really much else about it that I was able to find. So I think that these studies, considering, again, as I said, the technology that's around today and how prevalent these problems are, as I live with heart defects, I know a few people who have heart defects, the research should be progressed, just figuring out why they happen and if there's new ways we can prevent them.
Great. And there's another question from the Q&A that I think you were about to read. I'll read it out loud for everybody. Anonymous asked, what was the most challenging thing about your research process?
The most challenging thing would probably be finding the most updated information, especially during the pandemic, where it was a little bit harder. You just basically had your search engine and whatever you were finding on that. And I often found multiple studies, but they would all be from varying dates, or some of them I was like, I couldn't find a date for them. So that would be my most challenging thing, was just finding the most recent data that's available. And that's another, I don't know if maybe there are studies that are happening currently and they just haven't been released yet. None of the results are being published, but that would be the most challenging thing for me.
Cool. Thanks so much.
Thank you.
That's a great presentation, Noora.
Thank you.
Preethi, are you ready for your presentation up next? Awesome. I see Jen added you in. All right. I'll let you take it away. Oh yeah, and I can give you a reminder is that I'll rejoin as your 2-minute warning, and then I'll turn on my video again when it's going to wrap up. Sounds good.
Hi everyone, my name is Preethi Nalluru, and today I'll be presenting about language development and communication and neuroanatomical differences in children with ASD. A little bit about me is that I'm a senior at Evergreen Valley High School in San Jose, California. I wrote this review because I volunteer with autistic kids, and when I found out that autism is a developmental disorder that mainly affects social communication and behaviors, I really wanted to find out more about the development. So as an introduction to autism spectrum disorders, they are neurodevelopmental disorders characterized by deficits in social communication and restricted and repetitive behaviors. Approximately 1 in 54 children is diagnosed with ASD, and the diagnosis stems from abnormal development seen in regular wellness checkups. The average age of diagnosis is 4, while a diagnosis can be considered reliable at around age 2. The most common language disturbances are echolalia, which is repeating an expression word to word, and pronoun reversal, using I when used intended. Like the slide says, there's high heterogeneity, meaning that there's a high variety in the symptoms that children with autism experience. As you can see in this picture, there are various symptoms and various severities in the symptoms. There's often reduced language, and onset is delayed, as I'll tell you later. Children with autism often experience a reduced sensitivity to human voices. They show reduced responsiveness to vocal stimuli, even though they show responsiveness to non-vocal cues. They show reduced responsiveness to their name as well. Canonical babbling appears in typically developing children at around 10 months. Canonical babbling is a string of syllables that include a consonant and a vowel. Children with autism show low canonical babbling production and a low total vocalization rate. Typically developing children show earlier first words. Their earlier first words appear around 12 to 18 months, while it is delayed in children with autism. Earlier first words are important because they lead to better cognition, language skills, and adaptive skills later in life. 3-year-olds with autism show reduced initiations of interactions, less interrogative questions, and responses to parents. A little later on in life, children with autism show fewer speech-related vocalizations, and the parental responses are less dependent on the speech-relatedness of the vocalization. This could be due to the increased parental attention that children with autism receive. Children with autism also show a flatter trajectory with a steep growth later, even though that they show the same number of words in each grammatical category, like nouns and verbs. This shows that they struggle more with the social aspects of communication than they do with the rules of grammar. This picture here summarizes what I've said before, that they show reduced orientation to name, and that comes later, and canonical babbling and first words. Social communication is also important. If I, as a young child, point to a cup, my mom would say, "Oh, that's a cup." So then I would learn that that's a cup. So when the children with autism show a delay in the onset of gestures, it comes with a delay in the onset of language, like I mentioned on the previous slide. And children with autism show a delay in the onset of gestures and fewer gestures. They're also unable to take meaning from the direction of gaze, and they show fewer gaze shifts. During play, they show less gaze shifts between the person and the object, and they show reduced gaze shifts towards the person. So before I talk about neuroanatomical differences, I should probably talk a little bit about the brain. The Broca's area, which lies in the left frontal lobe, is important for speech production. And the Wernicke's area, which lies in the left temporal lobe, is important for speech comprehension. The arcuate fasciculus is a large bundle of nerve fibers that connects these two areas. Another thing I must mention is that it's the left side of the brain that is important for speech comprehension more than the right side. So in children with autism, they show less temporal activation and less left lateralization compared to controls. There's also less correlation in the activity between the left frontal and temporal activation. There's also increased cortical thickness in early childhood and more rapid decline in adolescence. This could be due to a delay in the processes like synaptic pruning, which remove the less important and excess connections in the brain. The relation between symptom severity and cortical thickness is strongest in the areas adjacent to the left arcuate fasciculus. Children with autism also show less superiotemporal sulcus activity. The superiotemporal sulcus is important for understanding eye gaze and also understanding the intention of another person's action, which is also impaired in children with ASD. So what I mentioned before is all important because it strengthens the watch and see approach. The watch and see approach is monitoring development over short periods of time and implementing intervention as soon as it's seen. So earlier intervention is important, right? Neuroanatomical differences may help diagnose ASD at earlier ages. Like I mentioned before, the average age of diagnosis is 4, while diagnosis can be considered relevant about 2. Well, that's 2 years of a later diagnosis. Early diagnosis is important because it means early intervention. And ASD is non-curable, which means that early intervention can help better the symptoms for the child experiencing the disorder. Like I mentioned before, the neuroanatomical differences that could contribute could be the cortical thickness. As I mentioned before, increased cortical thickness, and there's a rapid decline later on in life. This could be related to the flatter trajectory, the steep trajectory later on in life. There's also a high, I must end on a final disclaimer that there's a high heterogeneity in the symptoms that children with ASD experience. But whatever I mentioned before happens in general, and by no means is to everyone that by no means applies to everyone with ASD. Thank you for listening to my presentation. As I finished, I would also like to thank my mentor, Jacqueline Moscow, for helping me write this review. Do you guys have any questions? Oh, okay. So is there a numerical scale for autism, or it's more qualitative? The diagnosis is more qualitative, although the diagnosis is it's separated into level 1, level 2, and level 3 in both social communication and restricted repetitive behaviors. So it is more qualitative because it's a behavioral diagnosis. Are there any other questions? Yeah, I don't see any more questions. So thank you all for listening to my presentation.